Hey guys and gals, welcome to the very last concept in this chapter. Now, what I want you guys to do today is I'm going to teach you guys about something called gene expression. So, at the end of this lesson, I want you guys to be able to explain how organisms can regulate gene expression and how this can lead to different traits. So, I want you to be able to tell me, in order to do that, you have to do a couple things. First, you have to tell me the difference between eukaryotes and prokaryotes in terms of gene regulation and use the key terms in your explanation. You want to be able to explain how organisms can regulate their genes, and you want to be able to use all the key terms in this section effectively in your explanation, which is a little bit tricky because the key terms here are very weird. And here are your key terms: operon, operator, RNA interference, and differentiation. So, what is gene regulation? What is it about? Well, basically, it's just turning genes on and off. That's it. If you have a gene for something, you can turn the gene on and off. So for eukaryotes, it's possible. So eukaryotes like you and me and the students in the classroom and the rabbit Vanicula in Miss Kelly's room, those are all eukaryotes. And they're also able to make proteins faster and slower. So for them, gene regulation also has to do with that. So how does it work in prokaryotes? Well, prokaryotes are thankfully very simple, but I use simple with like big, massive quotations because it's not easy, guys, and I do apologize for that. The first thing that happens is you're going to have an operon. An operon is going to be a group of genes that are regulated together, and a group of genes that are on the same on and off switch. So in this picture right here, even though it's not labeled, it would be this part right here, the structural genes. That would be your operon. Basically, it's the protein you want to make. So think of the operon as the protein or proteins that you want to make if you happen to be a bacteria. The other one is called a promoter. Now, the promoter, hopefully you know this word because it's the same thing from the first section, and this is where the RNA polymerase is going to bind to. It's where it's going to attach and say, aha, I must make this. And finally, there's something called an operator. The operator, which can be pictured right here, is basically a place where something called a repressor can bind. A repressor can go to the operator and basically prevent the creation of this protein right here. What will happen is that if the RNA polymerase comes down here and binds onto the promoter, it's not going to be able to go this way because this big thing is in the way. So an operator basically acts like the off switch. If the operator goes to if the repressor goes to the operator, then done. It's not going to make that protein. So here's an example of an operon. This is called the lac operon for things like lactose. So basically milk, cheese, fun stuff like that. What happens here is that this is the gene in question. Here is a regulatory gene. What this regulatory gene does is that it's going to make the repressor. The repressor is going to bind to the operator, and it's going to keep RNA polymerase from actually activating. And right here would be your operon. It's going to be your protein. But since the repressor is there, it's not going to work. Now, how this gene works is that if lactose, so lactose being milk and milk sugar, if lactose, I'll put here LAC for lactose, if lactose comes in, it's going to bind to this operator right here. And when it binds to the operator, it's going to turn off the repressor. And then RNA polymerase is going to be able to attach here to the promoter, and bam, it's going to be able to make the enzyme to eat lactose. And when there's no more lactose in the cell, it's going to remove this right here, and the repressor is going to go back to the operon, or the operator, and you're done. This is basically how the bacteria regulate their genes. It is complicated, and for that I apologize. Hope this made sense. If not, come talk to me. Okay, eukaryotes. Ha 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 ha. They're a lot more complicated. Like crazy more complicated, unfortunately. they use something we call transcription factors. These are basically chemicals that can turn genes on and off, but they can also increase and decrease the speed of production. So that if for example, you like to drink lots of alcohol for some reason and you're underage, what can happen here is that if you drink lots of alcohol, then transcription factors are going to increase your body's production of chemicals that can deal with alcohol. And that's what it is. It's going to make transcription factors. And this right here is a very complicated picture of one, because really there are no simple transcription factors. They're all crazy complicated. Basically what happens is that a group of enzymes called activators are going to bind to genes, and the more of these activators there are, the faster this is going to go. And that's the basic picture. And trust me when I say it's a basic picture. But why do this? Why care? Why do eukaryotes do it? And it comes down to specialization. Basically, as you've probably noticed, eyeballs and skin are different. They don't do the same thing, do they? Well, how does that work? Because the problem is that your eyeball and your skin and your brain and your foot, they all have the exact same DNA. So how does the body know that it should make an eyeball on your head and make a foot on your foot area? Well, it does that with transcription factors. Basically, what's going to happen is that transcription factors are going to turn on and off genes depending upon where the cell's located. So if you have a cell in the eye region, the transcription factors are going to turn on the eye genes. If you have a cell in the heart region, the transcription factors are going to turn on the heart genes and turn off the brain genes. Because you don't want the brain genes on in the heart, you want the heart genes on. Here's a quick example, and this is coming from the area of genetic engineering, and it's something called RNAi, or RNA interference. RNA interference is just an example of a transcription factor. And there's the bell, because you can guess I'm still at school. So I'll let the bell do its thing. I'll sit here and talk for a second, because it's a very big distraction, and it's done. Okay, so the transcription factors, like RNAi, are going to be able to turn genes off. And what happens here is that RNAi is going to bind to DNA, and by doing that, it turns the whole process off. So in this example right here, you have infested roots versus healthy roots in tobacco plants. So what's happened here is that the scientists have used RNA interference, or RNAi, to turn off genes that would make it so that this plant can defend itself against this infestation. So they basically use science to create RNAi that can turn genes on and off. And your body can do this too, even with the help of scientists. Last part of here, guys, is a concept called differentiation. This is basically when a cell becomes different kinds of cells. So for example, heart and brain cells have the same DNA, but they look very different. They have a different structure. Differentiation is when a cell decides to become a new type of cell. So what happens here is that you have stem cells. These are basic cells that don't have much turned on, much turned off. They don't really know what they want to be. Think of them as like high school students. They have the knowledge to do a lot of things, but they don't really know what they want to do with themselves in life. So stem cells can differentiate. They can become muscle cells, blood cells, nerves, heart tissue, liver cells, and intestinal cells. They can become any kind of cell they want. They're basically cells that can do anything. But once they become that cell, so once a stem cell becomes a muscle cell, it's done. It can't go back to being a stem cell. At least not yet. And that's what differentiation is. And that's it, guys. I hope all this made sense. If you have any questions, come talk to me in class, and I will see you all in class.